Good morning, everyone. So, in the last class, we discussed about the complete gram-positive bacilli like Corynebacterium diphtheriae, Clostridiae, and also Bacillus anthracis. So, in today's class, we shall cover one of the important major aspect in bacteriology, that is Enterobacteriaceae, the family called as Enterobacteriaceae. One point what you have to remember is whatever bacteria we are going to discuss right from today for another one or two classes, so all these are gram negative bacilli. So the family Enterobacteriaceae includes all gram negative bacilli which are predominantly habitat having in the large intestine. So they are the predominant bacteria present in the large intestine, and all are gram negative bacilli. And look at the biochemical reactions what we presented there. All Enterobacteriaceae, they are oxidase negative. That means if you want to place a particular type of gram negative bacilli, it has to satisfy certain properties to have the taxonomical status under the family Enterobacteriaceae. So all are oxidase negative, all are catalase positive. Do remember the reagents used for catalase test and oxidase tests. All can reduce nitrates to nitrites, and test is called as nitrate reduction test. Some are fermenters, some are non-fermenters. That is very important point, and that is the key point. Some are fermenters and some are non-fermenters. So this fermentation property is attributed to the carbohydrate lactose. So we can elaborate them as some are lactose fermenters, some are non-lactose fermenters, and I will introduce another term called as late lactose fermenters, which can ferment the sugar lactose very slowly. Some are capsulated, some are non-capsulated. Some are motile and some are non-motile. If motile, motility is by means of peritrichous flagella. What do you mean by peritrichous flagella? Where the flagella are distributed entirely around the bacterial cell. Okay. Now, coming for the classification of the family Enterobacteriaceae, look at the first criteria what we presented. It is based on the fermentation of the sugar called as lactose. Whatever bacteria which are having the ability to ferment the sugar lactose, they produce pink colored colonies on the differential medium or the indicator medium. Indicator or differential solid medium. What is that? It is McConkey agar. On McConkey agar, they produce pink colored colonies because of fermentation of the sugar lactose, and hence they are called as lactose fermenters. So I presented two examples here. Typical lactose fermenters of Enterobacteriaceae are Escherichia coli and Klebsiella. Now, how to differentiate between Escherichia coli and Klebsiella, you may ask me, as both are lactose fermenters and both produce pink colored colonies. So it is very clear. I will show you the image also. Escherichia coli is it will produce pink colored flat dry colonies, except very few strains having a capsule like K1 strains, they produce slight mucoidness, whereas Klebsiella produces large sized dome shaped highly mucoid colonies because of the presence of a well defined capsule. Okay. Then late lactose fermenters, which can ferment the sugar lactose very slowly after 24 hours. It includes Citrobacter, one of the important species, and one species of the genus Shigella sonii. It is a late lactose fermenter. It is a PG NEET MDS entrance exam question. So 2 years back they asked this question. So please just revise the old question papers, and commonly asked Viva question also in your second year practical exams. Shigella sonii, it is a late lactose fermenter. Then non-lactose fermenters. So completely they do not ferment the sugar lactose at all, and they produce colorless colonies or pale colonies on McConkey agar. They do not produce any color, they do not ferment lactose, no acid production, nothing will be there. They produce colorless or pale colonies. They are called as non-lactose fermenters. So two very important examples here. You can see here, one is Shigella. So remember Shigella sonii, that species is late lactose fermenter, whereas other species of the genus Shigella, they are all non-lactose fermenters. So Shigella, it causes bacillary dysentery. Then another important one, Salmonella. Okay, Salmonella species causes typhoid fever or enteric fever. Okay. Next, scientific version of classification, taxonomical classification, that is called as a Bergey's manual Kaufman Edward Ewing classification. So according to this classification, so bacterial families are named as groups or families or tribes. They consist of genera, subgenera, species, and further species includes biotypes, serotypes, bacteriocin or colicin type. Important classification, and please remember this is one of the area of questioning either in your second year BDS exams or in the PG NEET MDS entrance exam. Okay, so very important area of questioning, and either you may get case study in your PG entrance or you may get some reasoning and assertion type of question also. Okay, so please have a concept. Coming for clinical infections, yes, already I mentioned this is one of the area of key point. It is a key area of this topic and a major area of questioning, especially in the entrance exam is concerned. So in general, the clinical infections caused by the members of Enterobacteriaceae includes diarrhea, urinary tract infections, pyogenic infections like wound infections, pyogenic meningitis, septicemias, bloodstream infections, etc. So in general, we are mentioning all these infections, and according to the nature of the bacteria, the infection differs. Okay, so urinary tract infections, diarrheas, pyogenic infections, septicemias, bloodstream infections. So major group of infections, the clinical spectra of infections. This is the image which shows the clear demarcation between the lactose fermenters and non-lactose fermenters when the collected clinical specimen is cultured on the differential medium, that is Mekong agar. So at one end you can see the pink colored colonies indicative of lactose fermentation. At the other end you can see the colorless colonies indicative of non-lactose fermenters or the pale colored colonies. Image based questions, so you have at least 2 to 3 from microbiology in the PG NEET MDS. Now coming for the description of first organism under the Enterobacteriaceae, that is Escherichia coli. So it includes gram-negative bacilli, Escherichia coli gram-negative, actually motile bacilli, very actually motile bacilli, and that is another important differentiating property between Escherichia coli and Klebsiella. Klebsiella is non-motile, Escherichia coli is actually motile. It is a typical lactose fermenter, catalase positive, oxidase negative. Now we are going to mention the 5 important diarrhegenic strains, the toxogenic strains of Escherichia coli. They are named as enteropathogenic Escherichia coli, enterotoxigenic Escherichia coli, enteroinvasive Escherichia coli, enterohemorrhagic Escherichia coli, enteroaggregative Escherichia coli. So these are the 5 different types of diarrhegenic Escherichia coli, the toxogenic strains: enteropathogenic, enterotoxigenic, enteroinvasive, enterohemorrhagic, enteroaggregative. First one, enteropathogenic. So few important points on enteropathogenic Escherichia coli. It causes diarrhea in infants and children, causes epidemic, but sporadic cases do occur. It is also called as enteroadherent Escherichia coli. Next, enterotoxigenic. So important strains include O6, O8, O15, O25 and 167. Look at the first point, that is a entrance exam question. So it causes traveler's diarrhea, organisms causing traveler's diarrhea. You please make a list. Okay, bacteria causing traveler's diarrhea, parasites causing traveler's diarrhea, viruses associated with traveler's diarrhea. So you have to make a list. Okay, it causes traveler's diarrhea and produces diarrhea when it gets attached to the mucosa. It produces either heat labile toxin or stable toxin. Next, enteroinvasive Escherichia coli. It is a atypical strain. Remember, it doesn't ferment lactose. It doesn't ferment lactose. Okay, it resembles one of the species of genus Shigella called as flexneri. It doesn't ferment lactose, but ferments dulcitol. But ferments a sugar called as dulcitol. And one of the important animal tests useful for the diagnosis of enteroinvasive Escherichia coli is Serini test. When the fresh culture strains of enteroinvasive Escherichia coli are inoculated into the animals like a rabbit, it produces keratoconjunctivitis. Now, one of the important property of enteroinvasive Escherichia coli is it has the ability to penetrate the cells due to the presence of plasmids. So these plasmids, they code for virulence marker antigen. Virulence marker antigen. Now, what are the major infections caused by Escherichia coli? Most predominant infection, the commonly isolated organism among urine samples collected routinely in hospital practice or in diagnostic setups is Escherichia coli associated with the UTI. Other than UTI, it causes wound infections, bloodstream infections, pyogenic meningitis, especially because of capsular K1 antigenic strains. And this K1 antigenic strain of Escherichia coli associated with pyogenic meningitis, it cross reacts with the group B polysaccharide of meningococci. Okay, this is the PG NEET MDS entrance exam 2016. It also causes primary bacterial peritonitis and also bone infections like osteomyelitis and also wound infections. And differentiating points between Klebsiella and Escherichia coli, biochemical reactions useful to identify. So imbic reactions for Escherichia coli, indole negative, indole positive, methyl red positive, Oaks prosecute negative, citrate negative. And remember, Escherichia coli doesn't ferment sucrose, it ferments other sugars, glucose and lactose, produces acid slant by acid but in the TSI medium. Whereas Klebsiella, imbic reactions minus minus plus plus produces urease, okay, and TSI it ferments all the sugars, acid slant by acid but. H2S producing bacteria in Enterobacteriaceae, Proteus, okay, Proteus. Clear? So I shall conclude the class. So we shall see in next class. So please do read and come. Okay, I will ask you questions. Thank you. And biochemical reactions, imbic reaction, indole production test, methyl red test, citrate utilization test, urease production test, TSI, triple sugar iron agar, I said mnemonic also. Okay, triple sugars, glucose, sucrose, lactose. Okay, that is GSL Medical College, Rajahmundry. And also Salmonella, Shigella, Citrobacter, mnemonic is SSC. Okay, your SSC 10th class public exam. Also question oral, that is important only due to clip features. Question reaction, that is a well, and also 2 marks question YK methods to inhibit question, how to inhibit swarming. That swarming growth plate we shall keep in the spotters for your second year practical exams. In spotters you have to identify it. If you are identifying correctly, you will get full marks, 1 out of 1. Okay, identify and write 1 or 2 relevant points. And whatever areas I mentioned regarding your MDS exam also, that is also very very important. Okay, so please read all these things and we shall see in your next class. Escherichia coli, introtoxinic Escherichia coli, 2 marks question, intro-invasive, swarming growth. So the significant bacteriuria under Escherichia coli, that is concept, more than 10 power 5 colony forming unit per ml, that is called as significant bacteriuria. That is a 2 marks question in your second year exam. Significant bacteriuria, more than 10 power 5 colony forming units per ml of the urine sample, that is concept of significant bacteriuria. And different strains of Escherichia coli, toxinic strain, invasive strain, pathogenic strain. Okay, and important properties of Escherichia coli, and pneumonia due to Klebsiella, Proteus, the swarming growth of Proteus. What are the properties of Enterobacteriaceae members? All are gram negative bacilli, all are catalase positive, oxidase negative, some are fermenters, some are non-fermenters, some are having capsule, some do not have capsule, some are motile, some are non-motile. If motile, motility is by means of peritrichous flagella. Okay, about excretion, remember you have. So that completes the class. So here today